Welcome to our meiosis video. Today we're going to talk about the purpose and process of meiosis, as well as ways it increases genetic variation. I actually want to start on the back of this big sheet, comparing mitosis and meiosis. This is really important. Mitosis is going to create body cells, and meiosis is going to create sex cells, also known as gametes, like sperm or egg. A body cell could be a skin, or a liver, or a brain cell, something like that. In mitosis, you start with one parent cell, and this parent cell is diploid, or 2N. And you end up with two daughter cells that are also diploid, or 2N. In meiosis, you start off with one parent cell that's diploid, also known as 2N, but you actually have two divisions, and you end up with four daughter cells, and each of these daughter cells are haploid, or N. Like in this example, you can see the parent cell in mitosis has 1, 2, 3, 4 chromosomes, and each daughter cell has 1, 2, 3, 4 chromosomes. So the daughter cells are identical to the parent cells. In meiosis, you start off with one diploid cell with 1, 2, 3, 4 chromosomes, and you end up with four daughter cells with only 2 chromosomes here. And each of these cells is very different because you have a lot of genetic variation that is introduced, which is very, very important here. We are talking about a human. A human would start off with one cell, one body cell, with 46 chromosomes. And at the end of mitosis, you would have two daughter cells, also with 46 chromosomes. Whereas in meiosis, you would start off with one parent cell with 46 chromosomes, and you would end up with four daughter cells, especially in the case of spermatogenesis, with 23 chromosomes. So let's look at the goal of meiosis. The goal of meiosis is it's used to prepare for sexual reproduction. So the goal of meiosis is to produce haploid gametes. Okay. To do that, we need to reduce the chromosome number by 1/2, and we also want to increase genetic variation. And why do we want to do these things? We want to do these things to prepare for sexual reproduction. So if you think about cats, okay, the mother and the father cat. First of all, cats have 38 chromosomes and 19 pairs of chromosomes. Okay, so a male cat is most of the body cells are 2N. There's 38 chromosomes and 19 pairs of chromosomes in both the mother and father cat and most of their cells. Okay. But if we want to make the next generation of cat, we need to do meiosis. So we do meiosis here, and I'm going to kind of highlight the processes so you see when the processes are occurring. We do meiosis here. The father cat creates sperm cells, and each of these sperm cells has 19 total chromosomes, and we have no pairs, which is why it's called N. Same thing with the egg. It's N. It has 19 total chromosomes. Okay? So that process that allows the mother and father to make these gametes, these sperm and egg cells, is called meiosis. So we've prepared for sexual reproduction. Next, we have fertilization. That's when the egg and sperm come together. So 19 chromosomes plus 19 chromosomes, and the zygote, or fertilized egg, now has 38 chromosomes again. So we're back up to 2N. So this process right here, this fertilization process, is a result of sexual reproduction. Meiosis prepares the sperm and the egg cells for sexual reproduction. Then sexual reproduction occurs when the sperm and the egg unites. We get a zygote or the fertilized egg. And then to grow the organism, we have mitosis and cell division. And this is for growth. During growth of this organism, two things are going to happen. Okay? We're going to have growth, but we're also going to have cell specialization. Each cell will change its form in order to have different functions. And that will be in our next unit on DNA, where we will discuss that in more detail. Let's look in more detail here about the process of meiosis. Okay? Basically, before meiosis, the DNA replicates 1 time. So, DNA replication occurs before meiosis. Then meiosis has 2 parts. It's kind of divided like right about here. We have meiosis 1 and we have meiosis 2. In meiosis 1, we have interphase, prophase 1, metaphase 1, anaphase 1, and like telophase 1 / cytokinesis. A lot of important things happen in meiosis. I said meiosis 2 there. That's actually meiosis 1. A lot of important things happen in meiosis 1. First of all, we have separation of homologous pairs. And this reduces the chromosome number from 2N to N. That actually happens in meiosis 1. Also, we have crossing over and independent assortment, which I'm going to put a major asterisk next to because these two increase genetic variation, and we'll talk about that in just a second. Crossing over happens here during prophase 1. It's when the homologous chromosomes exchange pieces of themselves. And then we also have independent assortment. Use a different color for that. Which is when the homologous chromosomes line up and separate from each other, and that happens in metaphase 1 and anaphase 1. So really the big deal in meiosis 1 is we go from diploid to haploid, and we increase genetic variation. Meiosis 2 is kind of just like mitosis. We are separating the sister chromatids, the 2 sides of the chromosome. So we don't change chromosome number really in meiosis 2. We just go from haploid to haploid. Okay? And in the end, we end up with 4 haploid cells, and we'll put a little asterisk next to that because there are some exceptions to that as well. So that's the process of meiosis. An important thing to note is there are really 2 types of meiosis, and we're going to talk specifically about humans right now. Okay? Oogenesis is the production of egg or ovum, and spermatogenesis is the production of sperm. Spermatogenesis is what I would refer to as a classic meiosis. Okay? You're making the cells that are going to become sperm cells by mitosis. So the meiosis part really starts here. And what happens is in the testes, there are these cells called primary spermatocytes, which are 2N. They undergo meiosis 1 to make secondary spermatocytes, so they go from 2N to N. They undergo meiosis 2 to make early spermatids. And then they differentiate, they become their sperm cells, 4 sperm cells that are haploid, they add the tails and that kind of thing. So this happens continuously, and you get 4 viable sperm. So it's spermatogenesis, it's continuous, and you produce 4 haploid viable sperm. Right? So spermatogenesis is what I would think of as classic meiosis. Oogenesis is also meiosis. However, there are some exceptions to kind of the continuity of it and how many egg cells are actually produced. So let's look at oogenesis. Oogenesis happens in your ovaries and actually happens before you're born. Okay? So again, meiosis starts here. Your primary oocytes, okay, which are these diploid cells, are present at birth, and they're stuck in prophase of meiosis 1. So when you're born, all of your egg cells are already in meiosis 1. Okay? Now, when you go through puberty and you have a menstrual cycle, okay, your egg cells finish meiosis 1 and start meiosis 2. Okay? But then they pause halfway in meiosis 2. It is not until fertilization actually occurs that this process of meiosis is finished in an egg cell. So this process of meiosis in egg cells is not continuous. Also, if you notice, there's an unequal division of the cytoplasm. So in meiosis 1, one of the egg cells gets most of the cytoplasm, the other egg cell gets almost no cytoplasm. So they both get the haploid amount of DNA, but this egg cell can't actually be fertilized. It's called a polar body. In the second division, once again, one of the egg cells gets all of the cytoplasm, the other egg cell gets nothing. This becomes a second polar body. So you actually only end up with one viable egg, okay, because of unequal distribution of cytoplasm. Okay, so it's a little bit different. It's good though because this huge egg cell is able to have mitochondria and endoplasmic reticulum and things like that it can give to the offspring. That's why you get 50.1% of your DNA from your maternal line, because you get all of your mitochondria from this huge maternal egg, and from the sperm you only get the 23 chromosomes that the sperm contains. So you get 23 chromosomes from the maternal line, 23 chromosomes from the paternal line, and then you get a little bit of extra DNA from the maternal line and the mitochondria. So that's the process of meiosis. I want to end this video with the importance of increasing genetic variation in meiosis. So increasing genetic variability in meiosis is extremely important. And there are 2 main ways this happens in meiosis, and 1 way this happens afterwards. So crossing over and independent assortment are both examples of ways to increase genetic variability during meiosis. Crossing over actually occurs during prophase 1 of meiosis 1. And what happens is these 2 homologous chromosomes, so you have 2 homologous chromosomes here. I'm going to kind of color code 1 in red, and I'm going to try to color code 1 in yellow. These homologous chromosomes line up next to each other, okay? And when they're lined up like this, when there's 4 chromatids together, they're called a tetrad. And what happens is they join together, okay? And it's called synapsis. They join together and they exchange genes so that when they separate from each other, one of the homologous chromosomes has much of its original information, but it has pieces from the other chromosome. And the same thing holds true. The same thing holds true for the other homologous chromosome. So basically, genes are exchanged between homologous chromosomes. And this exchange of genes increases genetic variation because now each of these chromatids are different. The other thing that can happen during meiosis is independent assortment. Independent assortment is the way the homologous chromosomes line up in metaphase 1, okay, and separate. So it's the how homologous chromosomes line up in metaphase 1 and metaphase 2 and separate. This can increase genetic variation in meiosis. Now, the last way to increase genetic variation is random fertilization, which occurs after meiosis during sexual reproduction. And basically all this is saying is like, it's a mystery which sperm and which egg are going to unite. And that's why there's genetic variation between the offspring. The last thing I want to talk about is in the back. What happens when meiosis goes wrong? When meiosis goes wrong, you can have something called non-disjunction. Non-disjunction is when the chromosomes do not separate. This can lead to a monosomy, which is a missing chromosome, or trisomy, which is an extra chromosome. Okay? And a good example of this would be something like this, which is Down syndrome. Instead of having 2 copies of chromosome 21, you have 3. So it's a trisomy. Other things that can happen are chromosomal mutations where parts of the chromosome are added or deleted. But we'll talk about that more later. Thanks so much. I hope you enjoyed the video.